In order to understand the lytic cycle and lysogenic cycle, we need to understand the structure and characteristics of viruses. Capsids are the protein shells that enclose the viral genome and are made out of thousands of capsomeres, which are protein subunits. The most common shapes of capsids are icosahedral, prolate, and helical. Next, the viral DNA that is enclosed in the capsid can either be DNA or RNA. The DNA viruses commonly use host RNA polymerase II to transcribe viral mRNAs, from which viral proteins are synthesized using the host machinery. On the other hand, RNA viruses use an enzyme called reverse transcriptase, where the viral RNA is converted into viral DNA. Some of the characteristics of viruses include that it's small, it does not have any organelles. Since it doesn't have organelles, it can't metabolize, and it can't replicate without a cellular host.
The lytic cycle is one of the two cycles of viral reproduction. This cycle is typically considered to be the main method of viral replication. This cycle results in the destruction of the infected cell and the release of new viruses. The first step is the attachment by the virus to the cell membrane, done by the tail fibers of the virus having proteins that are drawn to the host cell, and the virus attaches to the receptor sites. Next, a virus must enter the cell through the plasma membrane. Once through, it releases its genetic material into the cell. This genetic material can take the form of either DNA or RNA. During the viral gene replication stage, the virus's nucleic acid uses the host cell's machinery to make large amounts of viral components, such as the genetic material and the structural parts of the virus. mRNA production is regulated with the enzymes involved in modifying the host's DNA replication by RNA polymerase. In the middle phase, the virus's nucleic acid is produced. In the late phase, the structural proteins are produced, including those for the head and tail. Eventually, the viral components are made and assembled into complete viruses. Soon, the cell is filled with around 100 to 200 brand new viruses, and the cells will lyse. This gives the lytic cycle its name. An example of a virus that goes through the lytic cycle is E. coli. Recent science has discovered an interesting occurrence in nature. That is, not all viruses simply explode out of the cell. Some viruses undergo a budding process in which they bud out of the host cell, taking parts of their plasma membrane with them. In certain types of viruses, budding maturation may occur. This is when a virus has budded off its host cell, but interestingly, still can't immediately infect another cell, even though it has all the parts. First, it must mature inside the membrane bubble. The most well-known case of a virus that undergoes this process is rabies.
In the lysogenic cycle, the viral DNA is incorporated into the host's chromosome and replicates along with the host DNA. Once the viral DNA is incorporated into the host genome, the viral DNA is called a prophage. As a result, the virus will appear dormant, and the cell will continue to undergo mitosis and replicate its own DNA and the viral prophage. Eventually, there is a trigger that starts the lytic cycle and viral components start to form. Scientists are not totally sure what triggers the lytic cycle to start, but they think it's environmental factors like UV radiation or chemical factors. Finally, the cell will undergo lysis and the cycle will continue again. HIV and AIDS is one of the most well-known lysogenic viruses and can stay dormant for weeks months, and even decades. As a result, people can have the virus for years and never know that they had it, and can unknowingly spread it to others. One thing that is unique about HIV is that it also uses the budding maturation process as a camouflage coat to prevent it from being detected by the immune system, since the cell membranes contain familiar cell surface proteins, just like rabies. Overall, the lytic and lysogenic cycles slightly differ, but they eventually result in the replication of viruses.
Hey, Mrs. Brown, can we talk about viruses?
What, 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 I'm gonna find a host, only got DNA in my capsid. I'm hunting, looking for a receptor, viral replication.
Now, attaching the host like what up, I'm about to dock. Now I'm just a virus, I got an M plate to lock. Infecting cell is so dang easy, receptors like dang, that fit me perfectly. Injecting my DNA, describing it to RNA, cells in control except the ribosomes. Now I've got it all in charge of this machinery. Should've changed this using RNA polymerase. Because I need to replicate, rule and control it. About to go and make some alteration, changing up those host promoters, making new cell owner unknown and powerless. I'm attaching and inserting my host, and I'm replicating. I'm a virus boy. I'm gonna make more DNA. No, for real. I'm gonna start making structural proteins, common cold, and some influenza, tobacco mosaic virus that's infecting. I am a case of SARS. I gave a case of SARS. I gave a case of rabies. Then they give some MERS. Lysis your cells in a crisis. Michael Bay ain't got nothing on my bursting game. Lysis, new cell virus free to infect viral. Viral just be like, it's a lytic cycle.
I'm gonna find a host, only got DNA in my capsid. I'm hunting, looking for a receptor, viral replication. I'm gonna find a host, only got DNA in my capsid. I'm hunting, looking for a receptor, viral replication.
What you know about lysogenic replication? What you know about DNA integration? I'm circularizing that DNA. Virus DNA, part of another cell's genetics. You can thank me for that genetic information, cause right now I'm doing genetic incorporation. I'm in the cell, you can find me in the DNA. I'm not struggling with genetic additions. Your grandma, your auntie, your mama, your mammy, I'll infect your whole family. Extended family, I'll contaminate those others. The built-in viral DNA is hidden with the others. I kill one cell and then we'll stop all of the others. They'd be like, oh, that virus, that's not alright. I'm like, yo, that's the lysogenic cycle. Viral addition, let's do some normal reproduction. The lysogenic cycle, that's just some quality division. I call that getting cloned and replicated. I call that getting recreated. That DNA is viral though, and having it separate from the other strands in the cell is possible. Peeping, come back to the lytic cycle. Trying to stop me from thriving, man, you hella won't. Man, you hella won't.
Cells, finding hosts, yeah. I'm gonna find a host, only got DNA in my capsid. I'm hunting, looking for a receptor, viral replication. You have my DNA, I will replicate. I'm in this capsid coat, assembled by my cellular host. You have my DNA, I will replicate. I'm in this capsid coat, assembled by my cellular host. I'm gonna find a host, only got DNA in my capsid. I'm hunting, looking for a receptor, viral replication.